everyone. Thank you for coming here today. And thank you, Sarah and Gina, for setting this up. I'm very excited to talk about our core and the services that we provide for all your immune monitoring needs. So I just wanted to start with an overview of our core. So IAC, Immunotherapy Assessment Core, was established in December 2019. We are located at the 4th floor of Terry Building. And the mission of IAC is to facilitate basic and translational studies, aiming to dissect mechanisms of immunopathogenesis and explore novel disease-specific biomarkers with diagnostic and therapeutic applications. Additionally, we also provide training opportunities for students, postdoc, staff, and faculty to learn immune monitoring techniques. A unique thing about our core is that we have capabilities for full service. That is, we can take a project from start to finish. You could just drop off samples with us. For example, if you need to do a high-dimensional flow cytometry with our Cytec Aurora, we could just take your PBMCs that you have from your clinical trials or other places. We can stain them for you. We can do the panel design and all kind of project setup things, and then we can run it for you and give you data. So that was just an example. So we provide both self and staff-assisted services, however much or less you need. To introduce you to our team, we have very insightful leaders, faculty directors, Dr. Zhang and Dr. Lapool, and a very talented technician, Megan, and I myself as a core manager. We do have an open position, so if you're interested or might know anyone who will be interested to join our team, please reach out to us. So to go further, let me tell you about immune monitoring. What is it? In a broad sense, you can understand immune monitoring as a method to quantify the magnitude of immune responses after treatment or during the diagnosis of a disease. For example, let's see here. Let's say there is a patient who underwent transplant or got a cellular therapy or some kind of treatment, whether radiation or any kind of drug treatment. Then you would need to monitor the immune responses after those treatments. That can be done systematically with the samples like blood, urine. It can be local with organ biopsies, or it can be a whole body tracking or by imaging methods. So the systemic monitoring could be through different functional assays where you will take the cells and run it through ELISpot or some kind of functional killing assays, those kind of things. You could also run flow cytometry to get a deep immunoprofile of the picture of the whole sample, or you could run spectral or mass cytometry with side-off kind of techniques. You could run your cells through gene expression methods like bulk RNA-seq, single-cell RNA-seq, or real-time PCR methods. And then with organ biopsies, you can see what's happening locally with like proteomics methods, histology methods, and spatial imaging like geomics or metal-based imaging like Hyperion, or spatial transcriptomics like 10x single-cell genomics, or multiplex imaging platform like the Koya has Codex or Vectra3 that we have in our core. So in our core, we cover almost most of the techniques that you'll see further. What I'll do is I'll go through the techniques, and after each technique, I'll give you an example how the data generated in the core were used in the publications. So during the talk, you can interrupt me. Let me know if you have any questions. These are just some of the examples with the data that was generated in the core that was published, just to give you a glance of it. An overview of the services that are provided by the core. So far, we provide 55 different kind of services that range from self-service and staff-assisted. You can go to our website and find the rates for those. They are both, like if you are Northwestern user, you can get a base rate. If your PI is a cancer center member or SBDRC member, you can get a discounted rate. And we do provide services to the external academic or commercial people also. So starting with the technologies that we have in the core. First, we have the multiplex imaging platform that consists of a Leica Bond RX autostainer. So our autostainer is a fully automated research stainer where you could run 30 slides for staining simultaneously. It can be used for immunohistochemistry, FISH, or multiplexing, or any other kind of test that you would like. It even has protocols to be integrated with RNAscope. It can accelerate the test programs, and it's very efficient and very robust, gives very reproducible data. You can use FFPE sections or frozen sections. It is very good in preserving morphology and integrities of your precious tissue samples. The protocols that you use here are very flexible. You can customize them for antigen retrieval, different kind of staining, depending upon the applications that you need. So as I said, this autostainer we use in combination with multiplex imaging platform that we have from Akoya Biosciences. It's called Vectra 3. So let me give you an example of the workflow, how we use it here. So let's say you have a panel. You were interested in looking at PD-L1, CD8, FOXP3, CD68, PAN-CK markers with this following colors in your tumor tissue section. So what you'll do is you'll give us the tissue sections, we'll run it in this autostainer, and then can take the images in the Vectra 3 imaging platform. And then when you take the images, the software that is there in the Vectra 3 can unmix the images for different colors. And it has a machine learning software also, which can give you like a very quantified data at the end of your analysis. So to support this kind of technology, we have like a lot of validated immune markers, antibodies, and panels. And the cool thing is that it can detect like 6 markers plus DAPI. So altogether, you can do 7 colors. As I said, the data that you get from it is very robust and quantitative. I'll show you examples in the further slides. We do provide panel design and project setup support. So we kind of take it start to finish, as I told you before. Just to give you a hint, we have projects ongoing or completed for human tissue section that you see here. All of this kind, brain, skin, muscle, different kind of cancers, spleen, bones, etc., and also mouse tissue sections. So yeah, to give you examples of how we use this platform. So this is a mouse spleen frozen section, which was stained for CD3, CD4, CD8, CD19, and Ki67, and DAPI for nuclear staining. You could see that the resolution is so good, and you could see almost all of the markers that were stained, and it gives you a very clear picture of the spatial location of all of these markers. So to give you further examples from this paper, which use the data generated in the core. So in this paper, which talks about the ERK1 or 2 phosphorylation predicts survival following anti-PD-1 immunotherapy in recurrent glioblastoma. So the authors found out that the tumor patients which had high expression of phosphorylated ERK proteins had higher survival rate. And then they wanted to see which of the cells are producing the phosphorylated ERK proteins. So they use this platform. And since this platform can show you a very good expression of co-expression of different kind of markers, so they were able to find out that these cells or these markers were associated with the high expression of PERK, and you see that SOX2 positive cells has the highest expression of PERK, and to some extent TMEM 119 and CD163, which are microglial and macrophage markers respectively, were also expressing some PERK. So you can image here, and this data was quantified from these images. And similarly, they wanted to see differences between the SOX2 PRK co-expressing cells in high PRK and low PRK tumor cohorts. So that also they saw, like, you'll see high PRK in this high PRK expressing tumor cohort. Further, they wanted to see what is the distance between the SOX2 positive PRK positive cells from the myeloid cells like TMEM19 or CD63 cells. So you could see that the cohort that had high PRK had very short distance between these immune cells and the tumor cells. However, the cohort with low PRK and had very longer distance between the tumor cells and the myeloid cells, suggesting that in the high PRK samples, there's a good homogeneous mixing of these two kinds of cell types. So there's like a good immune activity there. So next technology is single cell immunophenotyping by Cytek Aurora. So the Cytek Aurora is a spectral cytometer. It's a very novel flow cytometer that expands the breadth of applications that were not available on the conventional cytometers. So the cytometer that we have in the core is a 3-laser one, and it can go up to 26 colors. It has very good autofluorescence extraction technique built up in the machine software. It can detect small particles also. We have validated mouse and human panels in the core for this on this machine. As usual, we provide all the panel design and project setup support and the sample staining support also, if you don't have resources or time to stain your samples. So just to go further in the technology, why it is so good, it can resolve the very similar spectra very beautifully. So as you can see here, in conventional cytometer, the emission peak is detected by the emission peak signals. You can see here, like APC and Alexa fluor 647 are very close. So there'll be a lot of overlap between these two. But with Cytek Aurora, since it looks at the spectral signature of this fluorophores, so it can distinguish them very clearly. So as you see, like just based on this tiny difference here and here, it can distinguish between these two colors and resolve very beautifully. So since you can do so many colors, you can do a lot of advanced analysis like t-SNEs, high dimensional plots like dimensionality reduction plots, heat maps with the data, and get a very quantitative data that you want. To give you an idea what kind of samples we have profiled in ISE so far. So we have done human and mouse PPMCs, a lot of clinical trial samples, human tumor samples, mouse tumor samples, mouse brain, spleen, and lymph node samples. To give you an example from this paper, that this data was generated in the core. This is a paper, Epigenetic Priming Enhances Anti-Tumor Immunity in Platinum-Resistant Ovarian Cancer. So here the authors wanted to show that how non-responders and responder cohorts, how that there was a difference between the immune landscape of those. And what they found was there was a high frequency of monocytes and DCs in the responder cohort than in the non-responder ones, which had a high frequency of non-classical monocytes that they see here also with the percentage of live cells that they calculated. So you see non-responders have low frequency of live cells, live DCs and monocytes, but have high frequencies of non-classical monocytes. They also found out that the non-responders had high expression of PD-1 and PD-L1. So this gives a very good idea why this responder cohort was very good in having an anti-tumor effect with the drug treatment. So next technique that we have in the core is a special transcriptomics and proteomics platform from NanoString. It's called Geomics. So they have panels for whole transcriptome atlas for mouse and human, and cancer transcriptome atlas for mouse and human both. So the way this technology works is you can have your tissue section. They can be FFPE, fixed frozen, or fresh frozen. They get stained with RNA or protein probes. And then with the morphology markers, as in the antibodies that you're interested in, the particular type of cell types. So they do have like 4 channels in the machine. So you could go up to 3 antibody markers, and 1 channel is for nuclear staining. So once you have your staining done, you go into the machine, you load your slide, then you can select the region of interest, depending upon whether your pathologist suggested you an interesting area, or when you image your slide, you see some interesting area, immune activity going on, depending upon how you want to select. They have like a range of selection options there. Once you have selected your ROIs, the machine is going to cleave the oligos, which are attached to your probes, which are photocleavable. So the oligos get cleaved, and they are collected in this kind of 96-well plates. So once they are collected, then we go further for library preparation, and then they are submitted for sequencing on the Illumina platform, and later then the data analyzed. So the NanoString Geomics also has an inbuilt software for software analysis, and you could also take the data and analyze with like your R packages or other kind of platforms that you want to analyze your data on. So really, it gives you a very good picture of the transcriptome in a spatial context, or proteome in a spatial context, because you can rightly see in which cells what kind of gene expression is there, or transcript expression is there. The projects that we are ongoing or completed in the core are for our human tissue section. We have done like projects for brain, ovarian cancer, glioblastoma, ADC. For mouse tissue section, we have tested it on brain and heart. To give you examples, so this was NRO1 data from NRO1 submitted from the Lipool lab. So here, they just wanted to validate the gene expression between 2 kinds of tissues. So first was human lung transplant tissues, and it was compared with the PDX, that is patient-derived xenograft, that was grafted in a mouse. So as you can see here, and this data is plotted between PMEL1 and PMEL2, meaning the PMEL1, like GP100, is a marker for this cancer. So this is plotted between this PDX tissue and the actual human original tissue. And same for the immune cells, we use CD45 marker. So that was also plotted between both of the samples. And you can see that like the gene expression was vastly similar between both types of tissue, validating the model is very relevant for further studies. And this is how the image will look in a geomics platform. So you would see that the green is GP100 and the red is CD45. I'm sorry, you cannot see the region of interest here. These are like tiny circles or squares here. Maybe in further slides, we can see it more clearly in a different example. So this is another example that I took from this publication that was available online. So this is a paper called Macrophage Infiltration Promotes Regrowth in Mixian Amplified Neuroblastoma After Chemotherapy. So here they use the Geomics platform to see the spatial transcriptomics data on their neuroblastoma samples. So they made use of CD45 markers for the immune cells and chromogranin A positive marker for the tumor cells. And they could clearly see a lot of activation of immune cells in the tumor samples. So they saw like both macrophages, both types, CD68, CD163, MHC class 2, dendritic cells, all this kind of cells. But they saw like more infiltration of CD163 in their samples. So by this, you can clearly see how you can dissect out the immune landscape or gene expression landscape between the pre and post-treatment tumor samples. So next technology that we have is a single cell transcriptomics platform from 10x Genomics. We have the Chromium controller. So we also have the capability to do the library preps for these samples. So we do have a bioanalyzer for quantification. PCR to do the amplification to make the libraries. Just an example of the quantification by the bioanalyzer. To give you an example, what kind of data you'll get with a 10x genomics platform. This is I took from a paper where authors studied the immune mechanism of protection after vaccine administration in a mouse model of vaccinia. So what they found out was that tissue resident memory cells adapted, like the CD8 tissue resident memory cells, they were adapted in different different parts of body or different tissues like lung and liver, and they express tissue-specific genes. So as you can see in this t-SNE plot, that they clustered very beautifully in this plot for lung and liver cells. And when you superimpose it with the tetramer positive cells, you'll see that very clearly how this tissue-specific genes clustered here in this t-SNE plot with their lung and liver tissue samples. And furthermore, they found out or dissected the tissue-specific genes, and you can see that these are very beautifully clustered here with their corresponding tissues. So this can really show you how you can tease out the minute details of gene expression at a single cell level in your tissue samples. Furthermore, we have a single cell secretomics platform from Isoplexis. It's called IsoLite. So it is a chip-based platform. So the chip kind of looks like this. As we know, ELISpot has been the gold standard for quantification of cytokines that is produced by T cells. So here they describe it as a single cell secreted proteins, or that are ELISpot on steroids, just because you can measure the cytokines produced by these cells in a real time. So it does a real-time ELISA on the chip that you can capture and quantify. So they make use of this metrics, what they call T cell polyfunctionality strength. This is essentially the metrics they use to calculate the percentage of cytokines and the number of cytokines that are produced by each single cell. So they have panels, both mouse and human, that have different range of cytokines, but mostly more than 30 cytokines that they can detect each time in parallel. And those can give you a very relevant data to correlate insights to improve immunotherapies. You can use samples here like single cells. So you'll use isocode chips if you're using single cells. The technology is also amenable to doing low volume bulk ELISAs. So you could also use serum samples, cell culture supernatant, CDC, and you would use CodePlex chips for those. So the sample volumes can be as low as 11 microliter. Just an example here, how the data would look like. So as you know, the cytokines or chemokines were detected in the range of 30s or 20s. So you'll see this here. This platform was utilized to see how CAR T cells are producing these cytokines and characterize them. And what the authors found out was there was like high production of granzyme B, IL-7, Brentus by CAR T cells. Those were CD8. And as you can see in the figure B, all of these four groups were further analyzed to show that the CAR T cells which had the transduced TCR receptors were they had like high polyfunctionality strength, meaning they produced more than one cytokine. So, So as compared to the untransduced CD4 T cells, they had like higher percentage of cytokines that were produced like in the multiples of 3 or 2 or 1. And same for CD8 T cells. The platform is also able to measure intracellular proteins. So this is an example where these are the four groups from from a sample where the M14 melanoma samples were treated with a nanoparticle. So these were the 4 groups, and you can see that the different kind of proteins that were measured fell into the categories of cell proliferation, DNA repair, or cytokine release. But the gist of this result is that what they found was CLIF-PARP, PE initiation factor 4E, and ribosomal PS6 drove functional heterogeneity. So this is also very cool, I think, that you can really tease out the intracellular proteins with this at a single cell level. So next we have a bulk secretomics platform also from Mesoscale Discovery. SQ120 is the machine. The sector imager. The features of this are it's like a very sensitive, low background, multiplex ELISA detection plate reader. It has a very dynamic range that enables biomarkers measures. You use very minimal dilution because you could do like very low volume for this panels. It is very adaptable to allow quantification of different kind of analytes. You can do single cytokines, or you can multiplex it with your choice of proteins that you want. They have like more than 200 proteins on their website that you can choose and select from and make the custom panels. So the principle it is based on is like it works like a sandwich ELISA, but it works on a principle of electrochemiluminescence detection. So there's a working electrode here, and there is a capture antibody that you put. Then you put your analyte, and then the detection antibody is sulfur tag labeled. That's why it provides very high amplification of your signal and low background. You can use it to quantify the cytokines or chemokines or other kind of proteins in the bodily fluids for samples like serum, cell culture supernatant, tears, and we have done some weird samples also. The panels, as I said, are available for mouse and humans and different kind of cytokines, chemokines, phosphorylated proteins. It is. To give an example from this paper, which was sting agonist loaded CD47 PDL1 targeting nanoparticles potentiate anti-tumor immunity and radiotherapy for glioblastoma. So here, this was a mouse. I think this was a CD26 mouse model of tumor where they injected with saline, or they treated it with this drug with radiotherapy. And they did the cytokine panel 1, which consists of like 10 different cytokines. What they found out was there was a significant difference between the CXCL10 and CCL2 production between the control mice and the drug-treated mice. And so both of these markers are T-cell activation markers. And then, as I said, this was a 10 protein panel, so they also were able to see CCL3, IL-30, CXCL2, IL-33, but there were no major differences between IL-33 and IL-30, I guess. So you can really get like good data from this multiplex kind of cytokine panels and find out at one go with low volumes that what kind of proteins are secreted in your samples, and they can be very useful for being a biomarker for that particular kind of disease. Next, the other services that are provided from the core are like GentleMax. If you want to do like a single cell suspension for further processing in the 10x genomics platform where you need single cells, or you need to lyse the tissues for RNA isolation, GentleMax is a very good equipment to do that because it's like it damages the tissue very little, and then you get high quality RNA or single cells from this. Lastly, we have like control rate freezer that you can use to freeze your samples at a controlled rate, and then it really preserves the viability of those cells. Service turnaround time for a core, I would say, is pretty decent. It can be 15 minutes to 3 days, depending upon what kind of technology or application you're using. We have capability to manage larger number of samples. You can do high throughput for many of the technologies. Technician time is available to develop custom protocols, and we try to provide customized services according to the user-specific needs for their projects. This is how you can get in touch with us. Our email, you can go to our website and you'll find all the information that you need about our core. If you need to discuss your projects, or if you have specific questions or inquiries, you can always set up a Zoom call with us, me or Megan, and we are very happy to help you. With this, I'll take any questions you have. Thank you.
You showed about this multiplex imaging,
Mm-hmm.
and so I know you have this really powerful probe also that can tell you the exact thing like you don't always have fresh. In the multiplex imaging, we were trying to intensities of different markers, but if you have the right panel of 7 or 8, could you kind of get like an immune profile, immune cell profile of your tumor from that? Like, will it enumerate like in a cell?
Right. Yes. Right. Yeah, it can.
Like something that could tell you.
Yeah. With context of the spatial location at the single cell resolution. With flow, you lose that spatial context, right? So here you can see. Yeah. 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 Yeah.
Yeah, like the single cell transcriptomics and the really nice spatial resolution. What about the single cell
proteomics?
Yeah.
So as I showed you, secretome is the one we have, isolite. It can tell you about intracellular proteomics and secretomics, but it's limited to like 30, 35 proteins. If you want to do proteomics, we do have like the nanostring geomics platform that I talked to you about. It's not really single cell unless it's like neurons or big cells, but it can get to a resolution of 20 cells, I would say. Exactly single cell, we don't have it, but we are trying to get this, not trying to get, but we really want to get this PhenoCycler fusion platform from Akoya. It's an extension of the Vectra 3 system that we have in the core. The differences and the upgrade is that it can detect up to 100 markers, more than 100 markers in like amounts of like 2 days of time. And it is at the single cell resolution. And they're coming up with a capability of RNA and protein on the both slides detection by the next year. So that would be very cool if we get that.
Everything is multi-omics
Yeah,
now.
yeah. It's like all omics.
That might be the next year.
We hope so. Fingers crossed. We are trying in different ways to do it, but let's see. Yeah. Thank you so much.